Hello students, very good evening. So third day MBBT, you're most welcome to everyone to be part of the session, and you can mail me also. We can go through it. I have one colleague of mine. We read through your journals, or you can also publish it. And after publishing, you can send it to us also, just to see that you can write it on your own and you can go ahead with it. So that will be fine in both cases. So somebody was India for PhD. Basically, students, yeah, on date and you can send everything. That's fine. Um, for PhD in India, basically you can check the professors also. If somebody, whatever you have done in your master thesis, you can check it and write an application to the professors itself. So ICER, all the ICER institutes, IITs, CSIR institutes, many more institutes they are still they're providing PhDs. DRDOs are providing PhDs. So basically, it depends upon your professor, your the course of studies you want to go for. You can also apply abroad. Just rightly direct to the professors that why you want to work under him or her, and what will you gain from that work, and what the other person will gain from you from the techniques that you have learned during master thesis and during your bachelor and master's course of work. So that's depend on that a lot. So today we will be dealing with recombinant DNA technologies, all the technologies related with the um, all the recombinant DNA technologies, all the PCRs, Sanger sequencing, um, DNA sequencing, Illumina, Soxola. So we will be dealing with that part today, and somehow we deal with the recombinant DNA technologies such as cloning vectors, cloning part with the how the cloning vector should be. So we'll be dealing with that part also. So you are allowed to modify the review topics. So I will be happy if you can have your own review topics for your studies, so that you can improvise the things as per your own idea. So that's no problem, no problem at all. So 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 Manjari, you can write it on your own. You can go ahead with this. Have some group people who could join you. Go with that. So further, any questions, students? Any questions? No, publishing is not compulsory, uh, but it's good for your career. You will learn how things are working in the field of academia at the just nascent stage of your BSc or MSc, whatever level you are. So it will be nice to have experience. So that's why I'm bringing this topic to everyone. Yeah, nutraceuticals. So you can go for in pharma field or something what you have done in your master thesis. You could go for that. I will try my best to answer your email, but sometimes I cannot reply. But I try to reply in these sessions. So I keep ping myself and try to reply here. So to publish, I will share with you one email soon. or I will share it here. There are some free journals we have find out. There you have to write to an editor with the journal, what have you written. If they are separate, then it's published. Simple as that. You don't have to pay anything. So I will tell you some free journals where you don't have to pay anything. So yeah, just give me 2 minutes. I'm coming back. Just give me 2 minutes. So students, let's start today's lecture. Um, Maybe we can start with this. Let's focus on this. So, a very warm welcome to today's topic of techniques in molecular biology. So we will be dealing something techniques such as analysis of nucleic acids, where we'll be dealing in the PCR, gel electrophoresis, some little bit about blotting technique, not in much details. Then we go for the gene expression analysis, real-time PCRs, microarrays. Then further we go for recombinant DNA technology, cellular sequencing and next generation sequencing, and method to study gene functions. Then further, we will go for cell culture methods and transgenic mice and knockout mice. So before we start, let me check if everything is fine with you students. Please thumbs up if, oh, we can't hear you at all. So my voice is gone or what? Mic testing, 1 2 3, 1 2 3. It's too low. Mhm. Maybe try to increase your volume. It's too low, too low, too low. Why is it too low? So volume is good. I try to fix it a bit from my side. Uh, is it fine so far? It's absolutely fine. Okay. So on that note, let's start the lectures today's. First technique that we will be dealing here is with the PCR, polymerase chain reaction, which means amplification of DNA. It can be used to amplify rare specific. We just take one sample, any DNA sample, and we increase its copies in millions. So by the end of the sequence, if we know the sequence, we can create the longer sequence of that. So PCR amplification of mutant alleles allows detection of human genetic diseases. DNA sequences can be amplified by PCR using in cloning as probes in forensic science. It was given by Gary Mullis, who got a Nobel Prize for Chemistry in 1993. So in the PCR mixture, you need buffer, template DNA, primers, nucleotides, DNA polymerase, and water. So all these six are the categories. All these categories are required with that. And further, this is how your PCR DNA is working out. This is how your PCR DNA is working out. This is how- So here you can see that there is a genomic DNA target sequence. Further, giving out you denaturation of heat by heat to denaturate your target sequence and separate the DNA strands. Then comes the annealing part in which you cool to allow primers to form hydrogen bonds with the ends of target sequence. Then in the extension, DNA polymerase adds to nucleotides 3' end of each primer, and then you keep increasing your nucleotides. And cycle 2, we repeat the same cycle, and you have 4 copies. Then you again repeat the same cycle, you yield 8 copies. By this, you can have amplified version of PCR, a lot of DNA. So that was a short way of telling it. Now let's go for uh, a virtual lab. PCR virtual lab. The human genome is made up of 3 billion chemical base pairs, and scientists want to isolate the specific segments of DNA within the vast amount of genetic material. So this is genetic material is so tiny, so they need many copies to have enough to work with. So this is your cell nucleus. In the nucleus you have a DNA. And you do PCR polymerase chain reaction that can generate 100 billion of identical copies of specific DNA sequence in matter of hours, and does not and thus requires no more than a test tube, few simple reagents, and source of heat. That's it. Three things that are required to increase your billions of copies of your DNA. So this is your hair follicle. So to perform reaction, you need DNA that has extracted from the cells. You don't need very much from the start of the reaction. For example, you can extract it from the blood, skin, saliva, or even from the hair follicle. To start, so this is the virtual lab now. To start the PCR, what you need is you need to extract DNA from that sample. So you need various PCR tubes. PCR works by heating and cooling the solutions over and over. So PCR tubes are designed for even heat distributions. So you extract your DNA that we did in the first workshop, the isolated DNA, and then release it into the PCR tube. And next, add primer 1 to the PCR tube, so that would attach to the copy to create the corresponding template. So primer 1 will go into the PCR tube. Now add primer 2, which will attach to the second site. We take primer 2 and we add to the PCR tube. Now add nucleotides to the PCR tube. So nucleotides like A, C, G, and T, so that they could make a DNA code, and these genetic building blocks used to create billions of DNA copies. So we will add some nucleotides to the PCR. So last, not the least, we will add the DNA polymerase, which will help to make the copies of it, matching nucleotides of DNA copies. This particular DNA polymerase has been specially selected to withstand the high heat of the PCR reaction. So take this one and it's gone. Now PCR tube is filled with all reaction components. Now we have to put them on thermocycler. This machine will precisely heat and cool the tube at specific times during the next hours, and these changes in temperature are crucial for reaction work. So drag the PCR tube to the thermocycler. This is thermocycler. Close it and then start. So what's happened inside the tube is, we will see now. So first you have, this is your target sequence for PCR. So inside the PCR tube, cycle has one begun. The thermocycler heats up to 95 degrees Celsius, that is around 203 degrees Fahrenheit, which is almost like boiling. At this temperature, DNA double helix separates and create two single-stranded molecules, like this. So we decrease the temperature to 50 degrees Celsius then. This temperature, single-stranded DNA molecules naturally attempt to pair up. However, there are many primers sequences than DNA in the tube. The primer crowd their way and lock into the target sequence before the strands could rejoin. So they are joined, so then DNA polymerase will join at the temperature of 72 degrees Celsius, and then it further starts to continue the reaction. So here your complementary DNA is produced against your DNA. So this is your cycle 1. This is your cycle 1 is complete in this way. Now comes again, we make 95 degrees Celsius, we separate them, then we make 50 degrees Celsius, then we go for 72 degrees Celsius. Click on the next button, and this way cycles keep going on. So during these 3 cycles, we have two strands that begin with primer 1 and end with primer 2. These are the DNA copies of just the segment of DNA we have targeted. So although there are only two desired fragments at this stage, as you continue with cycles, these products quickly become the majority. So cycle 4, so end of cycle 4, you have your target sequence, 8 fragments. By cycle 5, you have more, 20 fragments, 22 fragments. So by there will be 30 cycles. In 30 cycles, you will have billions of fragments that contains only your target sequence. That's 60 copies of longer length molecules, and you have now solutions nearly target whatever that you have targeted, you will get it there. So that's how the PCR is done. So how can you know these primers are complementary to the strand? So we create these primers via software to check to have specific that they could attach to that part. So we build them by ourselves. So there's a software you can build them by yourself. How do we ensure that molecules that have undergone annealing uh, how do we make sure that what's the question undergone annealing? So annealing is done at the temperature of, because this is all what we do. It's just I've told you that's the principal part. But once you do the PCR, uh, so this is the PCR protocol. So they are forward primer, reverse primer with DNA template. So the forward primer and reverse primer. So we create them very specifically to your sequence that you want to increase. So they will attach to that specific part. Like there is GCCT, and also on the other side is TCCC. So the other primers that will be corresponding to them. So they will create forward to create the same primers that you're looking for. That's how you control it in that way. So I will share you now. Uh, I can share the PPT already with you this time. It's okay. Maybe at the end of the workshop. But at the moment, I can share with you this PCR method. So just bend it. Where the rest of the part gone? Which part you mean? The rest of the DNA, it's in there. We are mixed together, but what we got at the end is maybe no, 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 no. Come on. No, no, no. So is the so as per the protocol, you take 2 microliter of your primers. So let's go to the next part. DNA molecules can be chemically synthesized also rather than using existing DNA pieces. New DNA can be designed and synthesized. So useful for in vitro generation of new gene structures, polylinker generations with MCS, primers and probes, generating new designer genomes. So all these things are possible with it. So this is your monomer 1 and monomer 2, and with coupling weak acids, so they are joined by the phosphate and oxygen bond. And then there is oxidation of I2, iodine, and detrotylation of the zinc chloride 2. So then you have your base 1 and the base 2. They are being attached, and repeat this process for monomer 3 or monomer 4, etc. So you will have a lot of oligonucleotides, DNA molecules to be attached, chemically synthesized. Then comes the gel electrophoresis, which resolve DNA fragments of different sizes. So you first restrict DNA restriction fragments, place mixture in the well of an agarose or polyacrylamide gel, and apply electric field. And then gel particle spores are done. They are moving along it. The molecules move through the pores in a gel at a rate inversely proportional to their chain length. There is a negative and positive end. Your shorter DNA will move faster, move down, and your longer DNA will remain up. So visualization of DNA samples separated by gel electrophoresis. So these are the clones, 1, 2, 3, 4, 5, and this is the molecular marker on the right hand side. You can see, and 1, 2, 3, 4, 5. So you can then check that there is 8 kilo base pair Dalton of DNA to be here in the sample clone 5, and you have 3 kilo base pair also present there, and the rest is 1 kilo base pair is present in the first clone, and also the 3 kilo base pair clone is present in mostly all of the clones, but in some that's the difference, and that's what makes your uh, DNA gel electrophoresis a unique technique. So if there are 5 people and you want to check with the forensic samples that what is the DNA by gel electrophoresis, how the DNA is made in that case. So you can compare and match the things. So this is how the DNA fingerprinting is done to check the crime scene blood reports. So now in the analysis of nucleic acids, we are done with the gel electrophoresis, we are done with the creating DNA molecules that chemically synthesized, and then polymerase chain reactions we are done. So we are done with the 2 techniques. Now third comes the blotting techniques, the analysis of nucleic acids. So it is a specific sequence present in a DNA sample. So is a specific sequence present in my DNA sample? Answer is yes. So we have specific. Everyone has specific DNA sequence, and everyone has its own role. So southern blotting, gel electrophoresis. Here you can see DNA that cleaves with the restriction enzymes. So there's a gel electrophoresis is happening. Then you transfer the samples for from the gel electrophoresis to the nitrocellulose membrane. Then you hybridize with the label DNA or RNA probe. Then you check it with the autoradiograph, how it looks like. These techniques we will deal in details. There's a day of southern blotting. So this is just to give you a hint to keep the continuity of today's lecture to have a good flow. And is the beta globin gene expressed? Is the mRNA present in total RNA sample? So answer is yes, expression increases over the time. So here the experiment is done. Again, gel electrophoresis. Here we have total RNA. Then you have a nitrocellulose membrane. The nitrocellulose membrane hybridized with the labeled DNA probe. And you can see with 48 hours and 96 hours, your beta globin gene expression mRNA is increased in 96 hours. So it's increasing. it is increasing the beta globin with the time. So yes, mRNA is present in our total RNA. So there is also yeah. So there is a real-time PCR for gene expression analysis. So this allows real-time monitoring of PCR amplifications products by the use of fluorescence. The first step is the reverse transcription of RNA into complementary DNA. Then either quantitative or semi-quantitative measurements are done, highly sensitive, used in research lab and clinical diagnostic labs. So this is the halogen tungsten lamp. Uh, in the first case is the halogen tungsten lamp and its excited filters are there. Then comes the intensifier. Then this is your plate sample plate downwards. Then it's making it uh, CCD lamp. So CD chip is watching it afterwards. So in plotting PCR in real time, so we can see that in different cell cycle number, this is our how much by 3 cycle number, 8 amount of DNA. So by the 34 or 30th cycle, we have this much amount of 1073741824 amount of DNA is produced. So it's increasing, the amount of DNA is increasing with cycles like in this phase. First very little, but at the end it's increasing very high. So per cycle number, the amount of DNA is increasing like this. Then further amplification curve. So this is how you calculate the output that is called relative fold change. This is how you can check application curve to determine the relative expression values. Um, so on the left side is the fluorescence, on the downward side is the cycle number. So initially, what you can see, there is a sample A and sample B, right? So there is sample A and sample B, right? So in the sample A, you have like this. You have by the 20 CTA. This is your from where your threshold is there. Sorry, like this one, CTA, this one, and this is for the CTB, the threshold value. And this is all is the log linear phase. This is the baseline and the exponential phase. Then it comes the linear phase. So to calculate it, it's E target divided to raise to power delta CT target control minus sample, then delta P divided by reference value control minus sample. So you have one reference value, you have one target value like sample A and sample B, and you calculate the delta CP. Delta CP is CTB minus CTA control minus sample. So I will play one video for the same now. Just go through it. So first, you prepare your reaction mixture, like a master mix of 25 microliter per sample, and you add forward primer to that. Uh, this is a virtual lab for real-time PCR. Then you add forward primer, 5 microliter per sample. Then you add reverse primer, that is 5 micrometer. So you can add reverse primer to that. Then you add, prepare the reaction mixture, Tapman probe, 5 microliter per sample. Then you allocate your mixture for 96 well plate. So you put them into the 96 well plate, your mixture. and then add complementary DNA sample to them, to your PCR tubes. Then cover the plate with microseal film. Then you place your tube in the PCR machine and run the program. So you analyze the results afterwards. These are the peaks that are coming. And then you will have the corresponding values of the 6 samples that you have added. Then you make a standard curve out of it. And then you can analyze with the formula automatically. So to take this RT-PCR, I will take you to the next part of COVID test that we are having. It is also done with the RT-PCR. So let's start this video, exciting video about that.
Acute respiratory syndrome coronavirus 2. When a person is infected, the most common symptoms include fever, cough, and shortness of breath. To start a test, the samples can be collected by a nasopharyngeal swab or an oropharyngeal swab. For nasopharyngeal specimen, the swab is inserted in the nostril and gently moved forward into the nasopharynx. Then it is rotated for a specified period time to collect secretions that contain the virus. Once the swabbing is applied, the swab is placed immediately into sterile tube containing a viral transport medium. The standard method of coronavirus testing is polymerase chain reaction, PCR, which is a method that used widely in molecular biology to make millions to billions of copies of a specific DNA fragment rapidly. Coronaviruses contain an extraordinarily long single-stranded RNA genome. To detect these viruses with PCR, RNA molecules must be converted into their complementary DNA sequences by reverse transcriptase. Then the newly synthesized DNA can be amplified by standard PCR procedures. This approach is universally known as RT-PCR. To perform this method, basically viral RNA should be extracted. Several RNA purification kits are available for convenient, fast and effective isolation. To extract the viral RNA by using commercial kit, the sample is first added into a microcentrifuge tube. Then it is mixed with a lysis buffer. This buffer is highly denaturing, and usually consists of phenol and guanidine isothiocyanate. Also, RNase inhibitors are usually present in the lysis buffer to ensure isolation of intact viral RNA. Once the lysis buffer is added, the tube is mixed by pulse vortexing and incubated at room temperature. Then the virus is lysed under the highly denaturing conditions provided by the lysis buffer. Once the sample is lysed, a purification procedure is carried out by using a spin column. The sample is loaded onto the spin column. Then a centrifugation is performed. This procedure is a solid phase extraction method, in which the stationary phase consists of a silica matrix. Under optimal salt and pH conditions, RNA molecules are bind to the silica gel membrane, and at the same time, protein and other contaminants are not retained. After centrifugation, the spin column is placed into a clean collection tube, and the filtrate is discarded. Then a wash buffer is added. The column is put in a centrifuge again, forcing the wash buffer through the membrane. This removes any remaining impurities from the membrane, leaving only the RNA bound to the silica gel. Once the sample is washed, the column is placed in a clean microcentrifuge tube, and an elution buffer is added. Then, a centrifugation is carried out, forcing the elution buffer through the membrane. The elution buffer removes the viral RNA from the spin column. And a purified RNA, which is free of protein, inhibitors, and other contaminants, is obtained. After the extraction of the viral RNA, the next step is the preparation of the reaction mixture for PCR amplification. In this step, a master mix is used, which is a premixed concentrated solution that consists of buffer, reverse transcriptase enzyme, nucleotides, forward primer, reverse primer, Tachman probe, and DNA polymerase. Finally, to complete this reaction mixture, the RNA template is added. The tube is mixed by pulse vortexing. Then the reaction mixture is loaded into a PCR plate, which generally contains 96 wells, allowing the analysis of several samples at the same time. Next, the plate is placed in a PCR machine, which is essentially a thermal cycler. Real-time RT-PCR is used for the detection of the new coronavirus 2019 by the amplification of target sequences in the RDRP gene, the E gene, and the N gene. The choice of the target gene depends on the primers and the probe sequences. The first step in RT-PCR is reverse transcription. The first strand complementary DNA synthesis is primed with the PCR reverse primer, which hybridizes to a complementary part of the virus RNA genome. Reverse transcriptase then adds DNA nucleotides onto the 3' end of the primer, synthesizing DNA complementary of the viral RNA. The temperature and duration of this step depend on the primer, the target RNA, and the reverse transcriptase used. Next, an initial denaturation step is applied, causing denaturation of the RNA-DNA hybrids. This step is required for the activation of DNA polymerase, and simultaneously the inactivation of reverse transcriptase. PCR consists of a series of thermal cycles, with each cycle consisting of denaturation, annealing, and extension steps. Denaturation step consists of heating the reaction chamber to 95 degrees Celsius, and it is used for denaturation of the double-stranded DNA template. In the next step, the reaction temperature is lowered to 58 degrees Celsius, allowing annealing of the forward primer to its complementary part of the single-stranded DNA template. The annealing temperature relies directly on length and composition of the primers. In the extension step, the DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand by adding free nucleotides from the reaction mixture that are complementary to the template in the 5' to 3' direction. The temperature at this step depends on the DNA polymerase used. After the first cycle, the double-stranded DNA target is obtained. Then, the denaturation of this double-stranded DNA is performed, yielding 2 single-stranded DNA molecules. In the next step, the reaction temperature is lowered, allowing annealing of the primers to each of the single-stranded DNA templates, and annealing of the Tachman probe to its complementary part of the target DNA. Tachman probe consists of a fluorophore covalently attached to the 5' end of the oligonucleotide probe. The fluorescence is emitted by the fluorophore when it is excited by the cycler's light source. Also, this probe consists of a quencher at the 3' end. The close proximity of the reporter to the quencher prevents detection of its fluorescence. In the extension step, DNA polymerase synthesizes new strands. When the polymerase reaches a Tachman probe, its endogenous 5' nuclease activity cleaves the probe, separating the dye from the quencher. With each cycle of PCR, more dye molecules are released, resulting in an increase in fluorescence intensity, proportional to the amount of amplicon synthesized. This method allows the estimation of the amount of a given sequence present in a sample. The number of double-stranded DNA pieces is doubled in each cycle, therefore, PCR can be used to analyze extremely small amounts of sample. For the measurement of the fluorescence signal, a tungsten halogen lamp, an excitation filter, mirrors, lens, an emission filter, and a charge-coupled device CCD camera are used. Filtered light from the lamp is reflected off mirror, passes through a condensing lens, and is focused into the center of each well. Then fluorescent light emitted from the wells reflects off the mirror, passes through an emission filter, and is detected by the CCD camera. In each PCR cycle, light from excited fluorifer can be detected by the CCD camera, which converts the light that it captures into digital data. This method is known as real-time PCR, which allows the monitoring of the progress of the PCR reaction as it occurs in real time.
So I hope things were clear, much better with this CCD of camera and all RT-PCR things that we have discussed now. Any further questions so far? How can you say that sample has only coronavirus without knowing the sequence of the virus? May it contain the rhinovirus? So, बच्चे, these RT-PCR techniques are developed basically for the primer that the sequence specifically to the coronavirus, so that we know that the coronavirus has these primer sequence, and if it is positive, then we see it. If it is not present, then we don't see it. So all the kits are prepared for the same reason. So could you be a bit louder? Is voice okay now? I was not aware. Maybe you have missed some important task. Okay, I hope my voice is fine so far. So let's continue. So that was something about RT-PCR and all these techniques. Further with the DNA microarray. So now comes the next part, DNA microanalysis. With the help of DNA microarrays, in which you have analyzing the genome-wide expression. So DNA microarrays consist of thousands of individual gene sequences bound to closely spaced regions on the surface of glass microscopes, slides, or synthesized sequence on a chip surface. So DNA microarrays allow the simultaneous analysis of expression of thousands of genes. The combination of DNA microarrays technology with genome sequencing projects enables the scientist to analyze the complete transcriptional program of an organism during the specific physiological response or development process. So you have fibroblast without serum, you have fibroblast with serum, and you isolate total mRNA out of it, then you reverse transcribe them to cDNA labeled with fluorescent dye. So there is on the left side green dye, on the right side is red dye. Then you mix hybridize to DNA microarray and you wash, measure the green and red fluorescence over each spot. So if a spot is green, expression of gene decreases in cells after serum addition. If the spot is red, expression of that gene increases. So this is how you see the each spot after your DNA microarray, like yellow, green, red, and each contains many copies of unlabeled DNA probe corresponding to part of one specific DNA. So the labeled complementary DNA hybridized to spots where there is sequence match. Red plus green gives you yellow color, identical expression level in both samples. So this is a bioinformatic analysis in silico that has been applied to the clusters of genes, could be mapped to the various biomolecular pathways. To understand it further, let's go through this.
This animation will demonstrate how DNA microarray experiments are performed. DNA microarrays, sometimes called DNA chips, reveal the expression of thousands of genes on a solid surface, such as a microscope slide. In this example, we'll use yeast as a model system to illustrate one use of microarrays. One common use of microarrays is to determine which genes are activated and which are repressed when two populations of cells are compared. Every gene is measured simultaneously. As an example, we'll compare what happens to yeast genes when cells are grown in aerobic versus anaerobic conditions. The cells grow and adjust which genes need to be activated or repressed in order to survive. Now it is time to isolate the mRNA from both populations of cells. The cells are spun in a centrifuge. Now that the cells have gathered in pellets, we remove the liquid, but not the cells. Next, it is time to extract the mRNA from the cells. When we add the extraction buffer, the mRNA is released into the solution. Next, we remove the RNA and place it in a fresh tube. Now, let's make the cDNA from the mRNA. Here we see 3 of the many mRNA molecules from each tube of cells. Each mRNA is converted into red or green colored cDNA. When the colored cDNA is made, the mRNA degrades. Then we combine the red and green cDNA, mixing both colors into a single tube. At last, it's time to look at the DNA microarray. In our experiment, a microarray or DNA chip contains about 6,000 spots. Each spot is a different yeast coding sequence from a different gene. Let's choose 3 spots at random to follow in detail. Each spot is made of DNA that can base pair with its complementary cDNA. Here are partial sequences from each of the 3 spots we are observing. Now, let's incubate the mixed cDNA with the DNA chip. For the sake of our example, we'll zoom in and show that some of the labeled cDNA have bound to the DNA in the spots and formed base pairings. Here we see green and red cDNA bound to this spot. Only red cDNA is bound to this spot. And only green cDNA bound to this other spot. In a real experiment, you would not see any of this detail. You would only see the original microarray. Now we must wash off the unbound cDNA to see what is bound to the microarray. Let's detect the bound cDNA so it can be visualized. We begin by placing the microscope slide containing the microarray inside a scanner. We'll examine the next phase of the process, keeping our focus on the 3 spots we've been following. First, a green laser scans the microarray. The resulting image is stored on a computer for later analysis. Now it's time for the red laser. This image is also stored on a computer for later analysis. Now, we move to the analysis phase. After we eject and safely store the microscope slide, we retrieve the red and green images from the computer and create a merged visualization. In the merged image, we see an aerobic gene labeled in green, an anaerobic gene labeled in red, and a gene labeled in yellow that was expressed in both aerobic and anaerobic conditions. This is one example of how DNA microarrays are used. In an actual experiment, quantitative analysis would be conducted on all 6,000 genes.
So that was about DNA microarrays. So with the different spots on the microarray plate, you can check whether your DNA was having the genes of aerobic, anaerobic, or the mixture of both. So to check it further, let me take you to the virtual lab of DNA microarrays. Okay. So this is a virtual biotechnique laboratory. We will talk through about DNA micro experiments. They are rather complex. Before you begin, let's have a solid understanding of the basics of DNA and gene expression. So virtual lab is broken into 3 chapters. Chapter 1 and 2 will give you some background, review a couple of important concepts that help to understand DNA microarray technology, and chapter 3 we will do the experiments. So you have a gene. So a genomic new science way back in 20th century, few years ago, scientists came up with many ways of study genes, mapping them, making mutations in them, cloning them, sequencing them, analyzing the proteins they encode. So lot of genes that you become really old. So typically scientists applied these kind of studies on one or maybe few genes at a time. but living things have a lot of genes. Humans have around 20,000 genes. It could take to investigate them one at a time, so it will take the whole life to do so. So that's why a microarray came. So wouldn't it be great if scientists could study many genes at the same time, or even better if they could learn about every gene in experiments in one single experiment? So basically, that's a bold ambition, but the kind of thinking was transformed the 20th century and into the 21st century of genomics. So genomics is a new word for new science. So genomics is a way to study many genes, thousands of genes, or even every gene in organisms all at once. Once proven, this task is known as DNA microarray. So you can study skin cells, muscle cells, pancreatic cells, erythroblast, melanocytes to see how powerful genomics can be. Let's make a closer look. Various different types with one another. So you have muscle cells, the nucleus, strands of DNAs are present, and gene 1, 2, 3, 4. So gene 2 and 3 are on, and the rest genes are off genes. So how do you know whether the gene is turned on? Well, one way is to determine if it is working, churning out molecules of messenger RNA, or the molecule gene is expressed or not. So your actin and myosin, they are on genes, the gene 2 and 3, so they will produce actin and myosin further with the help of mRNA. So these are your proteins. Whereas melanin and insulin is off, so they will not do anything. So that was happening in the muscle cells, where your actin and insulin and melanin is off, but your rest of the genes were on in that case. The same case will be not happening in the pancreatic islet cells, where your myosin and melanin will be off, but the rest of the genes will be on. So you can have a difference of genes present, and you can check whether the DNA with the help of DNA microarray using that point out the difference in the gene expression between two different cell types. So there are two different cell types, and we can check whether that gene is present or whether gene is not present by running that experiment on it. So this is nothing, just blah blah. This is a portion of thousands of spots on the microarray. Each spot represents one gene. Let's take a closer look. So microarray made up of thousands of spots. Each spots contain multiple copies of unique DNA sequence, which corresponds to a single gene. As we will see a little later on, this will be a handy tool for determining the difference between two cell types. So in this chapter 3, we are going to use the power of genomics to answer a very important question. What is the difference between healthy cell and cancer cell? So you might see, to answer this question, you should look at all the healthy cells and cancer cells under the microscopes and compare their appearance. That's one way doctors try to diagnose cancer. But cancer cells don't always look different from healthy cells, and comparing how cancer cells and normal cells looks different, that's where what we're gonna see it. So we're gonna see the genes. Gene 3 is broken in the cancer cell, and they get multiply with the same amount of gene, and because of gene 3 is broken, gone bad, many genes controls the way cell grows, divide and die. So when those genes stop working properly, cell growth can spin out of control, leading to tumor formation and cancer. But different kind of cancers are caused by different set of genes. So to able to diagnose, to treat cancer, it is better to know which genes cause the working abnormally. So here comes the microarray comes. So you collect your tissue, you isolate RNA out of it, then you isolate mRNA, make a labeled DNA copy, then apply that DNA, then scan the microarray, then you analyze the data. So we need microarray, simple tubes, scalpel, solvent solution, pipettes, vortex, microcentrifuge, column tubes, buffer solutions, green and label mixings, microarray washing solutions, microarray scanner, and workstations. So we're going to answer the question, what is the difference between your healthy cells and cancer cells in this case? Which genes were present in the cancer cells, which genes are off in the cancer cells, which lead to this difference. So we will compare it with the healthy cells. So this is the cancer tissue. Melanoma is happening on the cancer, and we're going to check with the same patient that how can we look at difference in the gene expression in the cells that have the same exact genetic blueprint. Only physicians, qualified scientists working with the physicians could be permitted to collect and use such tissues. But for this virtual lab, we will put on the virtual doctor caps. So our patients has arrived. Let's get started. So collection tube, let's grab it. We collect the cancerous tissue and we take the normal tissue. And then with the help of we can extract RNA by using the solvent. We can separate the DNA, protein and other cell components, leaving only RNA in the solution. So we add some solvent to the healthy tissue and also to the cancer tissue sample. Then you vortex them so that the tissue got mixed. Then you do the vortex the cancer tissue. Click on the lid to close the centrifuge. Then green button to turn it on. So you have healthy and cancer cells. So upper part would be the RNA and bottom the palette part would be protein, DNA, other stuff. Then you collect your samples, your RNA from the healthy sample and from the cancerous tissue sample. Then you isolate mRNA. So there will be mRNA, tRNA, ribosomal RNA, all RNAs will be present in our sample, but our task is to collect mRNA. So to do so, we have a poly A tail for the mRNA. So only mRNA reflects gene expression, so we will save the mRNA and get rid of the others. So you have close-up of single beads. These are the poly T attached to the beads like this. So you take the healthy samples which have mRNA, tRNA and RNA and you put into the column 1. From here you will have all the tRNA and ribosomal RNA to be dissolved downwards, but in the box you will have mRNA left. Again we repeat with the cancerous cells. So the poly tail will be attached to the beads, poly T tail, and mRNA will be attached to that part. So we add buffer, and now to remove your mRNA to the bottom. So now you add your green labeling mixture and red labeling mixture to these healthy and cancerous mRNA. So we add green into the healthy. So mRNA. So you have mRNA and polytail. So we have poly T trimer, reverse transcriptase is added, and we have labeled nucleotides. So labeling mixture contains, the green labeling mixture contains your poly T primers, which binds to the poly A tail, and they do the usually refer to the scientific community as oligo DT primers. And you have reverse transcriptase, an enzyme which will synthesize your complementary DNA against your RNA. Then you have labeled nucleotides, which will be incorporated into the new complementary DNA molecules. And nucleotide, these have been fluorescence molecules attached to them, so they will make a complementary DNA which has fluorescence light. So like this, your complementary DNA will be attached with the help of reverse transcriptase. Your complementary labeled cDNA is produced. And same goes in the cancerous cells. So good work. Our tubes no longer contain RNA, they contain complementary DNA now. And a healthy complementary cell DNA molecules are green and the cancerous cells are red. So that is what we apply to our microarray now. So two complementary strands are separated and rejoined. And two complementary strands from different sources can pair with one another, and this process is called hybridization. For the next experiments, we will take advantage of key property of DNA, that is hybridization. So two complementary DNA strands are mixed together. They will soon find and base pair with each other, forming a double-stranded DNA molecule. It doesn't matter where they came from, where they will do it. Scientists call this as hybridization, and it is the key of how DNA microarray works. Then you apply DNA stuck to our microarray and little piles of single-stranded DNA molecules. A single spot is contained, and then so this is your same DNA sequences here, and these are your different DNA sequences in that part of your microarray. So our computer database lists which gene is contained in each spot. So it will tell us that this spot has this gene, this spot has this gene. Now comes to add our sample to the microarray. And also the cancerous cells. So let's take a closer look now. We have what's happening on the surface of the slides. We have green one, red one, and also the red and green one hybridized one. So on this spots are the hybridized genes. So these are the healthy genes, complementary genes on this spot. These are the red cancerous hybridized genes, and there are some unhybridized label complementary DNA in the surface, and also some doesn't have any complementary DNA, so they didn't attach to any part, and some are together. So these spots, that what we see is red, green and yellow. Now we will wash the extra cDNA that does not bind to the complementary DNAs into our microarray, and then we check. Let's analyze our data under the scanner. So we scan our data into this microarray scanner. So finally we will scan the microarray to see where the green label complementary DNA from the healthy cells bound, to simplify things and examine the section of the microarray. Let's scan the green part. So these spots are the green parts that are transcribed into healthy cells. Now let's scan red part. So a lot of spots again are not green, but some we can see a lot of parts to be red now.
So these are from the cancer cells. And but how we can sort through the data to make sense of it? Microarray scanner will merge your red and green together. So please press now merge. So we will merge them. So now you can see that there are a lot of genes that are present in both healthy and cancerous cells. So some are still present, genes were present as a healthy cell here in this one, and these are the cancer cells, but most of the genes are present in the cancer and healthy cells. This is what this is showing. So here red and green spots merge together. Any spot that contain both red and green complementary DNA show up as yellow. A yellow spot contains genes that hybridize to both green and red cDNA, which means that genes are expressed both in cancer and healthy cells. Genes that show up as yellow spots probably aren't very interesting to us since their activity doesn't change when the cell becomes cancerous. But look at the red spots and the green spots. So red spots and the green spots are important to us. So red spots are the genes that are present specifically in the cancerous cells, and the green genes are specifically present in the healthy cells. So we can say that the cancerous cells are becoming because of these genes, and healthy cells are becoming because of these genes. So that's how DNA microarray helps us to differentiate between uh, different things. So red spot shows genes that produce more mRNA in the cancerous cells than in the healthy cells that turned up in cancerous cells. So, and the green spot shows the genes which turn down in the cancer cells. So you are a researcher, you have to study the genes increased level of activity in skin cancer cells. What color spots would you choose to study? So students, you want to check that which spot is high cancerous cells are happening. So which spot will you choose? The red spots, because these are the one that turned up in the cancer cells. So lot of red spots we can see. So there is a gene, they are the red on our microarray because they are controlled by a gene that has gone bad. So gene 426 is the one of the genes that are turned up in the cancer cells, and gene 426 is produces a protein product whose role is to turn down the expression of several other genes. So what other color spots that turned down by gene 4263? So what color are the spots? So green spot, yes, turns down the sets of the microarray. So gene 621 is a normally turned on the skin cells. In our cancer samples, the gene is defective, while the gene still produces mRNA, and the defective prevents the translation into proteins. What color gene is 6219? It's a yellow ones represented by the microarray. So 621 gene in the healthy cells is producing protein, whereas the gene is not turned on in the cancerous cells, this one, so it's not producing any protein in that case. So scientists rely on these techniques to call protein expression analysis to tell them if the mRNA is being translated into protein or not. So that was something about uh, protein expression and DNA microarray, how we do it. So let's take a break on this point. Let me have some water, glass of water, and we meet in 10 minutes or so at 6:20. Or 6:25. Let's have a 10 minutes break. So next, let's continue, students. Yeah, very basic questions. So I will send all the videos for sure. So to know that whether it's a cancerous cells or healthy cells, we will take them from the cancer or healthy cells actually. So we will take the specimen, make sure cancerous cells will be on the skin cell. They will be growing abruptly. You can see melanoma growing on the skin. So that's how we can differentiate between them. So if you do add both, then it become hybridized. You will have both cancer and healthy cells. Then when you add into the same, you can add red and green both colors also. Then you put them onto the microarray. Then you will have a very diverse kind of results. But that will be not giving you a output as a good output at the end. So please take care of that also. So mRNA lifespan is too short. How can we manage that? So that's how you have to do all these reactions at the room temperature. If you do at the room temperature, it will be not. You will get your results. That's for sure. I have done that by myself. So that will be not problem. That will be not a problem. So I can share these PPTs, sure. So all the videos are included within them. So I don't have to share the videos separately, but I can share you this DNA microarray. Not on this, on this. Yeah. So I will also share the first day we did this genetics. So some students were asking about plant DNA extraction protocol. So that I will share also that one. So we are done with this part. So now comes that investigating the genes cloning part with the biotechnology approaches, which is quite important. So because applied therapeutic cloning, so producing big amount of identical copies of DNA of interest, you can wait a second, wait a second. So you can have identical copies of DNA of interest, the gain of functions that you're looking for, expression of genes in other gene organisms to obtain large amount of protein, to investigate function of a protein in a cell culture model animal, generating functional DNA molecules for gene therapy in gene-related diseases. Genetically modified organisms could be produced with the help of these. So by deleted, like knockout, added transgenic mice or change mutant genes for the purpose of generating improved agriculture crops and resistance against the vermin or have increased content of healthy persons, healthy protein. Generating transgenic mice, in which we have medical research. Transgenic animals are produced, are important for producing the disease and possible treatments. In farming, generation of healthy animals with improved production potential. So all these are possible with the help of GMOs. So DNA cloning with plasmid vectors. So your recombinant DNA technology enables to produce large number of identical DNA molecules. Clones are typically generated by the placing a DNA fragments of interest into vector DNA molecule, which can replicate in bacterial host cells. So when a single vector containing a single DNA fragment is introduced into host cell, large number of fragments are reproduced along with the vector. So two common vectors are E. coli, plasmid vectors, and bacteriophage vectors. So plasmids are electrochromosomal self-replicating DNA molecules. So like HINT3, SPH, PSTL, SAL, XBL. So these could be your various multiple cloning sites could be possible here. And this is your plasmid cloning vector with ampicillin R as an antibiotic resistance site, and this is origin of replication. So these three sites are quite important in your cloning vector. So general procedure for cloning with recombinant DNA fragments: once you have a plasmid vector, other side is a DNA fragment to be cloned. Then you have enzymatically insert DNA into the plasmid vectors, and then you have a recombinant plasmid. Then you mix E. coli cells with the plasmids in presence of calcium chloride, culture on the nutrient medium, place on the containing ampicillin. So bacterial chromosomes will transform them and they will survive. Cells do not take up the plasmids, die on the ampicillin plate, which do not survive, they die in the ampicillin plate itself. So then independent plasmid replication happen. You have more grown plasmid inside your bacteria, then cell multiplies and cloning of each cell containing copies of same recombinant plasmids at the end. So plasmids cloning permits isolation of DNA fragments from the complex mixtures. So you have plasmid vectors, DNA fragments to be cloned, enzymatically insert DNA fragments into plasmid vectors. Then you have transform E. coli cells and select for ampicillin resistance colonies. So here you can see each colony represents one clone. So like this, one clone are present for each DNA fragments. So your restriction enzymes cut DNA molecules as specific sequence. So restriction enzymes are site-specific DNA generating molecules. Until now, more than 600 naturally occurring restriction enzymes have been identified, and they recognize 4 to 6 base pairs or sequence patterns. Restriction cuts are most often asymmetric, generating single-stranded overhangs. Restriction cuts are also less frequently symmetric in that case. So restriction enzymes looks like this. You have restriction enzymes E. coli, so it cuts at the specific part leading to have sticky ends. You have sticky ends at the end. And restriction enzymes recognized sites are usually palindromic, so they are palindromic at the end. Example of palindromes are like ENA, auto, radar, kayak. So whatever you read it from the left or the right, they mean the same. So these are the various examples of restriction enzymes: BAM H1, SAU3A, E. coli 1, HINT3, SMI, SM1. So their source of the microorganisms are like this, and they cut the recognition sites like this, and they produce specifically sticky or blunt ends like that. So blunt end is basically cut by the small, so they're cutting in the middle and leading to have no sticky ends at the end. So restriction fragments with complementary sticky ends are ligated easily. So you can see here TTAA, then OH to be on top, then you have DNA2. So it has 3 different types of complementary DNA which could ligate to that DNA1. But which one is more prominently gonna add is gonna be the A1, the first one, like this there. The complementary and base pairs and unpaired fragments B and C. Then you have a DNA ligase, 2 ATP, 2 AMP, 2 PPI, and you have a recombinant DNA at the end. So let's watch a small video about it, about restriction enzyme.
Plasmids can be good cloning vectors because they carry an origin of replication and are therefore able to replicate independently within a cell. Most plasmids used as vectors also encode some type of selectable marker, such as the gene for resistance to ampicillin. If the host cells are ampicillin sensitive, the only host cells that can grow on a medium containing ampicillin are those that have taken up the plasmid. Vectors must also have a small sequence of base pairs that can be recognized by a restriction enzyme. When this enzyme opens the circular plasmid, foreign DNA can be incorporated. When the plasmid vector and foreign DNA are both cut with the same restriction enzyme and mixed together, not all molecules will join to form recombinants. Some vector molecules will reanneal without incorporating foreign DNA. To identify cells that contain plasmids that have incorporated foreign DNA, a second marker gene is needed on the vector. This second marker contains the restriction enzyme site within its nucleotide sequence. If foreign DNA is inserted, the second marker is inactivated. This is referred to as insertional inactivation. A common second marker is the LAC-Z gene, which codes for the enzyme beta-galactosidase. Beta-galactosidase can cleave a colorless chemical called XGAL to form a blue compound. Therefore, colonies of cells that harbor the intact vector but no new recombinant DNA can make beta-galactosidase and form a blue color in the presence of XGAL. However, colonies that contain new recombinant DNA cannot make beta-galactosidase and are white.
So that's how
The difference of beta galactidase, which is a blue clone, and white clone is the no beta galactidase. This is how you clone your DNA in that sense. So now comes the polylinkers, that insertion of restriction fragments into the plasmid vectors. So here you can see that the sequence of polylinker, like E. coli, SARC, Carpnan, Thrines, SMI. So we have a sequence of polylinker, then insertion of E. coli restriction enzymes. So these polylinkers, they are being removed with the help of E. coli, and also the genomic DNA that you want to insert, they are also cut it out with the help of E. coli. Then they are inserted into that with the help of DNA ligase and ATP, and then you have a recombinant plasmid at the end. Same way, cloning the transcriptome, complementary DNA libraries are prepared from the isolated mRNA. So here you can see that there is oligo DT Petrix. So here they are attached, a mixture of cytoplasmic RNA mixed under the hybridization conditions, and mainly the mRNA are attached on top, and then wash away ribosomal RNA and tRNA. Elute column in a low salt buffer, then you have purified mRNA out of it. And same way, I think it's not required to go into this way. So identifying, analyzing and sequencing cloned DNA. The most common approach to identify specific clone involves a screening of library hybridization with radioactively labeled DNA or RNA probe. So labeled nucleotide probes are also used to identify specific nucleotide sequence in a complex mixture of DNA or mRNA. So sequencing, direct sequencing is also often applied, single nucleotide sequencing, large scale sequencing. That is sequencing whole libraries of DNA or complementary DNA. Further, DNA sequencing with the Sanger dioxy method. Let me show you one video related to Sanger method that will be more easy to go for, which will make sense to go back.
So that was about Sanger deoxy method. So here you can see that all the primers that are corresponding to ATPs, DGTPs, DTTPs, DCTPs, they do go denaturate and separate daughter strands by electrophoresis, and you can check specifically the DNA sequences as of now, as of the things. So now next comes the after the Sanger, which is also commonly used, but nowadays it's very old technique to go for. So most of the people, they are using um, next generation sequencing. So in this case, in the next generation sequencing, you have very technology platforms like Roche, Illumina, Applied Biosystem, Helicos, so which have various parameters you have to check, like cost, read length, speed, accuracy, preparation time while doing so. So in the years so far, 2004 is the 1 month worth output of the next generation sequencing machine. So by 2004, there's a drastic increase in the of your number of base pairs per billion for your next generation sequencing. So there are mainly three types of next generation sequencing: Roche, Illumina, and Solid. And we are discussing about today mainly to this point. Then we will switch it off for today. So let's focus on these three different sequencing. In the chemistry point of view, it's based on the Pyrrhus sequencing. Illumina is a polymerase based, Solid is the ligation based. Amplification, it's an impulsion PCR. It's a bridge amplification in this, and this here is the impulsion PCR is also happening here. Pair ends with the yes 3 kilo base pairs, 200 base pairs, 3 kilo base pairs in this case. The run is around for 100 mega base pairs, the 1300 mega base pairs, 3000 mega base pairs. Time run is 7 hours, 7 hours, 4 days, 5 days respectively. And cost is around 8,000 in this case, 439, 8,950. Here is a 17,470, so the most expensive one. But per base pair, it's the most expensive, 84.39 and 5.97 and 5.81. So somehow Illumina seems to be fine to go for, but it takes a lot of time for your work. So Roche, Illumina, and Solid, they have the different way of working. Let's go through this video.
Next-generation sequencing, or NGS, is a powerful platform that has enabled the sequencing of thousands to millions of DNA molecules simultaneously. This powerful tool is revolutionizing fields such as personalized medicine, genetic diseases, and clinical diagnostics by offering a high-throughput option with the capability to sequence multiple individuals at the same time. Sanger sequencing, first developed in the 1900s, is the gold standard for DNA sequencing, and it is still used today extensively for routine sequencing applications and to validate NGS data. It utilizes a high-fidelity DNA-dependent polymerase to generate a complementary copy to a single-stranded DNA template. In each reaction, a single primer, complementary to the template, initiates a DNA synthesis reaction from its 3' end. Deoxynucleotides, or simply nucleotides, are added one after the other in a template-dependent manner. Each reaction also contains a mixture of 4 dideoxynucleotides, one for each DNA base. These dideoxynucleotides resemble the DNA monomers enough to allow incorporation into the growing strand. However, they differ from natural deoxynucleotides in 2 ways. 1. They lack a 3' hydroxyl group, which is required for further DNA extension, resulting in chain termination once incorporated in the DNA molecule. And 2. Each dideoxynucleotide has a unique fluorescent dye attached to it, allowing for automatic detection of the DNA sequence. As a result, many copies of different length DNA fragments are generated in each reaction, terminated at all of the nucleotide positions of the template molecule by one of the dideoxynucleotides. The reaction mixtures are loaded on the sequencing machine, either manually onto slab gels or automatically with capillaries, and are electrophoresed to separate the DNA molecules by size. The DNA sequence is read through the fluorescent emission of the dideoxynucleotide as it flows through the gel. Modern-day Sanger sequencing instruments use capillary-based automated electrophoresis, which typically analyzes 8 to 96 sequencing reactions simultaneously. Next-generation sequencing systems have been introduced in the past decade that allow for massively parallel sequencing reactions. These systems are capable of analyzing millions or even billions of sequencing reactions at the same time. Although different machines have been developed with various differing technical details, they all share some common features. 1. Sample preparation. All next-generation sequencing platforms require a library obtained either by amplification or ligation with custom adapter sequences. 2. Sequencing machines. Each library fragment is amplified on a solid surface with covalently attached DNA linkers that hybridize the library adapters. This amplification creates clusters of DNA, each originating from a single library fragment. Each cluster will act as an individual sequencing reaction. And 3. Data output. Each machine provides the raw data at the end of the sequencing run. This raw data is a collection of DNA sequences that were generated at each cluster. The differences between the different next-generation sequencing platforms lie mainly in the technical details of the sequencing reaction and can be categorized in 4 groups: pyrosequencing, sequencing by synthesis, sequencing by ligation, and ion semiconductor sequencing. In pyrosequencing, the sequencing reaction is monitored through the release of a pyrophosphate during each nucleotide incorporation. The released pyrophosphate is used in a series of chemical reactions resulting in the generation of light. Light emission is detected by a camera which records the appropriate sequences of the cluster. The sequencing proceeds by incubating one base at a time, measuring the light emission, if any, degrading the unincorporated bases, and then the addition of another base. This technology is capable of generating large read lengths, much comparable to the read length of Sanger sequencing. However, high reagent cost and high error rate over strings of 6 or more homopolymers have reduced its applications. For more details on the technical aspect of this technology, please visit our knowledge base at the link provided in the description below. Sequencing by synthesis utilizes the step-by-step incorporation of reversibly fluorescent and terminated nucleotides for DNA sequencing and is used by the Illumina NGS platforms. All 4 nucleotides are added to the sequencing chip at the same time, and after nucleotide incorporation, the remaining DNA bases are washed away. The fluorescent signal is read at each cluster and recorded. Both the fluorescent molecule and the terminator group are then cleaved and washed away. This process is repeated until the sequencing reaction is complete. This system is able to overcome the disadvantages of the pyrosequencing system by only incorporating a single nucleotide at a time. However, as the sequencing reaction proceeds, the error rate of the machine also increases. This is due to incomplete removal of the fluorescent signal, which leads to higher background noise levels. Our NGS, an introduction knowledge base, provides more technical details about this technology. Sequencing by ligation is different from the other 2 methods since it does not utilize a DNA polymerase to incorporate nucleotides. Instead, it relies on 16 octamer oligonucleotide probes, each with one of 4 fluorescent dyes attached to its 5' end that are ligated to one another. Each octamer consists of 2 probe-specific bases and 6 degenerate bases. The sequencing reaction commences by binding of the primer to the adapter sequence and then hybridization of the appropriate probe. This hybridization of the probe is guided by the 2 probe-specific bases and upon annealing is ligated to the primer sequence through a DNA ligase. Unbound oligonucleotides are washed away, then the signal is detected and recorded. After that, the fluorescent signal, along with the last 3 bases of the octamer probe, are cleaved, and then the next cycle commences. After approximately 7 cycles of ligation, the DNA strand is denatured and another sequencing primer, offset by 1 base from the previous primer, is used to repeat these steps. In total, 5 sequencing primers are used. The major disadvantage of this technology is the very short sequencing reads generated. Ion semiconductor sequencing utilizes the release of hydrogen ions during the sequencing reaction to detect the sequence of a cluster. Each cluster is located directly above a semiconductor transistor, which is capable of detecting changes in the pH of the solution. During nucleotide incorporation, a single hydrogen ion is released into the solution and it is detected by the semiconductor. The sequencing reaction itself proceeds similarly to pyrosequencing, but at a fraction of the cost. Please view our knowledge base for further details on ion semiconductor sequencing and the sequencing by ligation techniques. In order to be able to showcase and compare the different technical aspects of each of the above technologies, the number of coverage that each run generates when sequencing the whole human, mouse, Arabidopsis thaliana, and E. coli genomes are calculated and presented here. The presented data is based on the most powerful machines of each technology. Further details can be found on our knowledge base. For whole genome sequencing data to be useful, a minimum of 30 times coverage is required. As it can be seen, the pyrosequencing method is only able to sequence the E. coli genome at enough coverage to result in valid data. The sequencing by synthesis method, which is the most popular method currently on the market, is able to generate hundreds of coverage per run. In fact, with this machine, it is possible to sequence 15 individuals within 3.5 days. The sequencing by ligation method also generates enough coverage for all genomes to be used. However, it isn't capable of generating nearly as much output as the Illumina HiSeq machines. The ion proton machine is used mostly in clinical settings because it is able to provide a sufficient size output within 2 hours. ABM offers a wide range of next-generation sequencing services. These include whole genome sequencing, exome sequencing, RNA sequencing, disease panels, lane rentals, and much more. To be able to access our services, please visit our website at www.abmgood.com, and from there, click on the NGS Sequencing Services link. This will load our NGS service webpage, which details all of our available services. Clicking on a service of interest will showcase the technical details, pricing, and bioinformatics solutions that are related to that particular service. Please leave your questions and comments below, and we will answer them as soon as possible. For more information, please visit our knowledge base at the link provided below. Thank you for watching.
So students, I hope this was clear for today's lecture. Okay, we will finish till here for today. From tomorrow onwards, we go to the next lectures. So I will share these slides tomorrow only, so not today. And so you can send me your assignments also tomorrow, so that you will be. I will send you the slides tomorrow after the lecture's over, because our lectures are still left. So I will keep continuing with them. So we are done with today. And or shall I send you the files today itself? What do you say? Shall I send it today itself? Because you will not study them tomorrow with me if I send it you today. Okay, I send it today itself. So it is being sent. It's a big file, quite big file. So it will take some time. Good. So any questions, students? Send me tomorrow your assignments, because our topic is not finished today. Send me tomorrow. So yeah, Sangar is not so important at the moment. So we have discussed in details which sequences are important. And one more thing, please subscribe to the channel by clicking on the subscribe button down. So this will really help us to make you more interesting slides. So you are around 125 students. So if you all make me subscribe this channel, so this will really help me to grow my YouTube channel and to start my YouTube family with all of you. So it's a humble request. After these lectures, if you can subscribe to my channel, I really appreciate that. Yeah, Adobe Flash is giving problem nowadays, but it will be fine with the time. So they will have some compromise with it. So I've already sent you the files. It's being sent. It will take some time to be sent today. So we are done with the 36 slides. So we will start with 37 slide tomorrow. Yeah, all clear? Alles klar? So I have already sent you everything now. Please go through them. So on that note, I will say goodbye to all of you. Thank you very much. It was a pleasure having class with you today of MBBT, and hope to see you more exciting works. And please, 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 humble request, please subscribe to the channel. Those whoever is watching it on live, just click subscribe button. Like and share, we will do it later, but first subscribe is important to grow it. Okay, how will you open those links without Adobe Flash? Bacha, you can open it actually. You have to just go into the settings to make your Adobe working. Assignments are not compulsory. All is good. Yeah, great, great. Thank you so much. Arigato gozaimasu. In Japanese, maybe in their accent, arigato gozaimasu, like this. Dijinkyu, maybe. Oh, it's new way of saying thank you. Thank you, students. Wish you all a pleasant evening, nice learning. May your all dreams come true. All the best in whatever endeavor you are going through. My best wishes with you. Take care. Uh, which functional group of isoflavin makes it more polar? Great, thank you. I have to check through it. This question. Sorry, I can't answer it. Please ping me if I can answer it personally. I can check. Okay. Somebody know the question's answers? Which functional group of isoflavin flavon makes it more polar? Okay, in Polish. Yeah, Germany and Poland, they are together. So students, I have my another class soon, so I need some break. Proteomics. So I have to leave now. I see you tomorrow, same time, same place with more exciting videos. In meantime, please, please, please subscribe to the channel, please. Thank you. Oh.